everyone, and welcome to today's webinar titled DNA Polymerases at Work, Finding Your PCR Champion. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher. For more information about Thermo Fisher Scientific, please go to www.thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. In addition to utilizing the live chat feature, we encourage you to participate in 3 poll questions throughout the speaker presentation. Click on your answer when the poll questions are displayed. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Marija Navickaitė, Product Manager of Platinum DNA Polymerases and Phusion DNA Polymerases at Thermo Fisher. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Marija, you may now begin your presentation.
Thank you, Sydney, for the introduction, and welcome everyone to today's webinar. So this is a brief overview of the webinar today. First, I will talk about basic principles and components of PCR, as well as major PCR applications. Then we will talk about evolution of DNA polymerases technology, how did first PCR look like, and which technological improvements enable PCR to develop into a technique that, as we know it today. We will also briefly cover what we can expect from PCR in the future. So PCR, or polymerase chain reaction, is a molecular biology method to amplify DNA even from a single copy to millions or billions of them. Nowadays, we see PCR as a fairly simple and very well-developed technique. And this is because PCR was invented more than 40 years ago in 1983 by Dr. Kary Mullis. Only 10 years later, in 1993, he already won a Nobel Prize in chemistry for this invention. It has been only 10 years when the discovery of PCR was already recognized as a very important scientific innovation with a great potential to make a difference in the future. Now let's take a look at basic components and basic principles of PCR. PCR is a doubling reaction, which means that in each cycle we have twice as much of PCR product than in the cycle before. If we start with the 1 DNA molecule, after 1 cycle, we will have 2 molecules. After 2 cycles, we will have 4 molecules, and so on. After only 30 PCR cycles, we will have a billion copies of the initial DNA molecule. And that's the real power of PCR, starting with very small amount of DNA and just in 30 cycles amplify it to such a high extent that allows it to be used in further analysis. Now, amplified sequence can be analyzed using different methods, and the choice of the method is really going to depend on type of PCR application that you are planning to do. In the endpoint PCR, the amplicon is analyzed by running an agarose gel. This is a good approach if you want to look at the presence and the size of the PCR product, or if you want to purify it for any other applications. If you want to quantify the amount of initial DNA, then the alternative approach is qPCR, or quantitative PCR, where the product is quantified at every cycle. And then digital PCR is another version. In digital PCR, sample is separated into large number of individual partitions, and number of target DNA molecules is then quantified directly. PCR is being used in a very wide variety of applications. For example, it is used in basic research for cloning or sequencing. It is also now widely used in medical diagnostics, for example, for genetic testing. And actually, the first medical test where PCR was used was to detect a mutation that caused sickle cell anemia. It is now also used to determine mutations in the oncogenes, because depending on the mutation, patients are prescribed with different drugs that work effectively for them, and also for detection of infectious agents like HIV, COVID, tuberculosis, and others. PCR is also used in forensics for human identification, as well as agriculture, and really many, many other applications. Now, looking into components of PCR and how it actually works. So here are essential components of the PCR reaction. So first, the DNA template, a sample DNA that contains the target sequence. Primers are short, 20 to 30 nucleotide long synthetic single-strand DNA sequences that bind complementary to the DNA sequence of interest. Then there is DNA polymerase. That's the enzyme, which is the key to PCR. It uses the single-stranded DNA as a template to synthesize the new complementary DNA strand. And DNTPs, or deoxyribonucleotides, they are used by DNA polymerase to build the new DNA strand. The reaction also includes reaction buffer and many other cofactors, including magnesium ions that are essential for DNA amplification. PCR is a three-step reaction. So it's denaturation, annealing, and extension. So first step is denaturation. In this step, PCR reaction mix is heated to a very high temperatures between 94 and 98 degrees Celsius in order to separate two template DNA strands and produce single-stranded DNA molecules. In the annealing step, the temperature is lowered from 55 to 72 degrees Celsius, allowing annealing of the primers to the single-stranded DNA template. Annealing temperature of primers needs to be calculated for every primer pair and varies depending on the size of the primer, its homology to the target DNA, and some other additional factors. There are enzymes actually that do not require annealing temperature calculation, and we will talk about that in detail a bit later. And then finally, there is extension step. At this step, DNA polymerase synthesizes the new DNA strand complementary to the DNA template by adding DNTPs. The temperature of this step depends on the DNA polymerase used, because different polymerases have different optimal polymerization temperature, but in most cases it is between 68 and 72 degrees. So one PCR cycle is composed of these three steps, and during PCR cycle, we have synthesis of two identical DNA strands. PCR reactions today, however, can also be done in two steps because annealing and extension steps can be combined if primary annealing temperature is close to the extension temperature for the DNA polymerase used.
All right. As a reminder, we would really appreciate your participation today by answering a few of our polling questions. To record your answers, click on the option on the screen. Our first polling question is, would you like to speak with a technical specialist and get a quote for thermal cycler for your research? I will give you a few moments to answer the question. All right, Maria, I will go ahead and hand it back over to you.
Okay, thank you, Sydney. Now, as I've mentioned before, one of the key components in PCR is the enzyme. And DNA polymerases, they have several activities. So first, the 5' to 3' polymerase activity is responsible for elongation of the primer that is annealed to the single-strand DNA. and the formation of the new strand in a 5' to 3' direction. Some DNA polymerases also have 3' to 5' exons or the proofreading activity. With this activity, DNA polymerases can verify whether every added nucleotide is correct. Incorrectly added nucleotide will be cleaved off and correct one incorporated. And based on these activities, there are two large groups of DNA polymerases. Non-proofreaders, these are enzymes that do not contain the proofreading activity and are often used in standard PCR applications. Proofreaders are the enzymes with proofreading activity and therefore are more accurate and used in high-fidelity PCR. DNA polymerases do not only differ by the presence of proofreading activity, they also differ by thermostability. Some are very thermostable, others are not. They also differ in processivity and speed. Some are very fast, used in fast PCR. Some are slow, resulting in very long PCR cycling times. They also differ by the ability to tolerate presence of inhibitors in the reaction. So when deciding and choosing which DNA polymerase to use, it is very important to consider all these characteristics. One needs to have in mind that specific PCR applications require use of DNA polymerase with certain properties. PCR, as we know it today, is very easy and convenient method. You can now quickly and accurately amplify a piece of DNA in a very small reaction volumes and use convenient PCR thermocyclers. That, however, was not always the case. 40 years ago, PCR was very tedious and time-consuming and required use of water baths and manual timers. So let's take a look and see how this technique evolved in the last several decades into modern PCR as we know it today. So let's take a look how were the first PCR reactions performed back in the 1980s. Back then, there were no PCR thermocyclers. In order to have 3 different temperatures for 3 PCR steps, you had to use 3 different water baths with different temperatures, and tubes were manually transferred from one water bath to the other. Also, time was manually controlled for each step. First PCR reactions were done with E. coli DNA polymerase, which was highly thermolabile and was destroyed at every denaturation step. So after each denaturation step, a fresh DNA polymerase had to be added into the reaction. Due to this, very large amounts of the DNA polymerase were needed for each PCR experiment, and this was, of course, very expensive. With this approach, only a few reactions could be performed at any one time, and the whole process was very tedious and time-consuming. Most important task at that time was to find a thermostable DNA polymerase so that it wouldn't have to be added at each cycle. Indeed, when we look into PCR evolution, improvements were mostly coming from PCR enzymes, and this was, of course, done in parallel by development of PCR instruments as well. And the first major breakthrough in PCR was replacement of E. coli DNA polymerase with thermostable DNA polymerase from Thermus aquaticus in early 1980s. Thermus aquaticus is thermophilic bacteria, which was discovered in geyser of Yellowstone National Park. Because this organism lives at high temperatures, it was expected that enzymes is isolated from this organism would be very thermostable as well. And this was true. DNA polymerase isolated from the organism was able to work at temperatures between 75 and 80 degrees and was named TAC. It had a half-life of 40 minutes at 95 degrees, so around 50 cycles under standard PCR conditions, and was therefore an ideal alternative of E. coli DNA polymerase. Use of TAC in PCR simplified this technique significantly because TAC could tolerate very high temperatures. It was no longer required to add a fresh enzyme during each cycle. This also allowed for development of automated thermocyclers. Recognizing its importance, the journal Science awarded Taq polymerase the first molecule of the year, and first paper describing the use of Taq in PCR became the most cited publication for several years in a row. Taq polymerase has 5' to 3' polymerase catalytic activity. It does not have proofreading activity. Taq also has the 5' to 3' exonuclease activity that removes nucleotides in front of the growing strand, which is useful in some PCR applications, such as probe-based qPCR. And Taq can effectively amplify DNA fragments up to 5 kilobases at speed of 1 kb per minute. Now, you should have in mind that there are differences in performance between different Taq polymerases, and depending on application, you should choose the enzyme carefully. Here, just to show you one example, 6 different Taq polymerases were compared, named A to F. 6 fragments were amplified from 160 base pairs to 75 kilobase size. First, what you can see is that all these Taqs are giving different product yields. With some Taq polymerases, we see bands of very high intensity. However, this is also usually followed by non-specific amplification. Then we also see differences in the ability of these enzymes to amplify the longest amplicon, 7.5 kb. Just a couple of enzymes were able to amplify this long fragment. So if you need high yields of your PCR product and or need to amplify amplicons bigger than 2 kilobases, you have to choose your enzyme carefully because not all Taq polymerases perform the same. Now, although Taq polymerase was a dramatic improvement over E. coli polymerase for PCR, it still had several drawbacks. Most important one would be that it is not a proofreading enzyme. Taq can incorporate incorrect nucleotides. Therefore, error rate of Taq is quite high. If you amplify 3 kilobase amplicon with Taq after 30 cycles, you can expect to have about 4 mistakes in every clone. So Taq is very prone to introducing errors, and because of that, nowadays it is used only for applications that do not require high fidelity, such as high-throughput screening, genotyping, sequence detection, or colony PCR. In all these applications, one does not need to know the actual sequence. The fragment will not be used for cloning. You just need to see whether your fragment of interest is there or not. Already at the end of 1980s, researchers were aware that there is a need for more accurate and robust polymerase, so the search for a new enzyme continued. So let's briefly look at what proofreading activity is. All enzymes possess the 5' to 3' polymerase activity that synthesizes new DNA strand. All polymerases make mistakes from time to time, which can lead to wrong data interpretation in some applications. Now, proofreading enzymes have the 3' to 5' exonuclease activity that enables polymerase to spot the mistake and correct it. The enzyme excises the incorrect nucleotide, incorporates correct one, and synthesis continues. Yeah, so just a reminder, there are 2 broad groups of DNA polymerases, non-proofreaders and proofreaders. The first group, non-proofreaders, lack the 3' to 5' exonuclease activity, so they are a good choice for low-fidelity standard PCR, where it does not matter if the PCR product contains any errors, mutations, and only yes or no answer is required. The second group, proofreaders, are DNA polymerase of choice if a DNA fragment needs to be amplified with high accuracy so that it doesn't contain any errors. While they all show a higher fidelity than a non-proofreader like the standard Taq, the difference between the proofreaders themselves is how much more accurate they are. While some have just 9 times higher fidelity. Newer proofreaders, for example, Phusion plus DNA polymerase, are 100 times more accurate compared to Taq polymerase. So the next breakthrough was discovery of PFU polymerase. PFU was isolated from Archaea, Pyrococcus furiosus, which was found on the Italian island. Pyrococcus furiosus is a hypothermophilic archaea that grows at astonishing 100 degrees Celsius, so its enzymes are extremely thermostable. Optimal temperature of PFU DNA polymerase is between 70 and 103 degrees. It remains 95% active after 2 hours of incubation at 95. Also, this polymerase has both 5' to 3' polymerase activity and 3' to 5' exonuclease activity. It can effectively amplify fragments up to 5 kb. It is, however, a very slow enzyme and needs about 2 minutes to amplify 1 kilobase. So PFU, unlike Taq, has a proofreading activity, meaning it can correct errors and therefore has a much lower error rate. 3 kb fragment amplified with PFU will have 1 error in 4 clones after 30 cycles of PCR. At that time, when it was discovered, PFU was most accurate DNA polymerase and was therefore used in many applications that required higher fidelity, including cloning, sequencing, and mutagenesis experiments. Today, as we will see later, we have even more accurate DNA polymerases that work even better in high-fidelity applications. And then the next big improvement in PCR was use of hot start technology, which significantly improved PCR reaction specificity as well as sensitivity. No matter which polymerase is used, often you see not only expected amplicons, but also non-specific amplification. These non-specific amplifications can significantly affect PCR reaction, its sensitivity and yields. In this example here, we see that the more of non-specific amplification we have, the smaller the yield of the target fragment is. And why do these non-specific amplifications occur? Well, there can be several reasons. The primers may not be designed properly, and they may target some other DNA sequence as well in the template. Maybe the cycling conditions are not optimal. Each DNA polymerase has its own recommendations for cycling conditions. So make sure cycling conditions that are being used are optimal for that enzyme. Long cycling steps can sometimes result in non-specific amplification as well. But it can also be that DNA polymerase is active during PCR reaction setup, and during this stage amplify non-specific products. Hot start technology avoids non-specific amplification by inactivating DNA polymerase at lower temperature during reaction setup and activates it only at elevated temperatures when conditions for primer annealing are more stringent. Now, first, hot start methods were manual methods and were implemented already in late 1980s. These methods involved simply heating up the PCR reaction without the DNA polymerase to more than 90 degrees, and then cooling to 60 to 70 degrees before adding the polymerase. This method was effective but tedious and carried the risk of cross-contamination of samples. Today we have numerous different methods. Most of them are now based on inactivation of some component of PCR reaction. DNA polymerase can be inactivated in many ways, including antibodies, chemical modifications, aptamers, affibodies, and others. So with hot start PCR, we can remove non-specific amplification, increase yields, as well as sensitivity of PCR. Hot start PCR is therefore used in all applications requiring very high sensitivity, and this would be in cases when you want to use a sample with low concentrations. Hot start enzymes simplify reaction setup, which can now be done at room temperature. And also it is critical to use hot start enzymes for high throughput screening experiments when robotics are involved. Now there are two properties of DNA polymerases that enhance their performance. That is processivity and start. So hot start enzyme is inhibited at room temperature and becomes activated during the denaturation step of PCR. The mechanism contributes to specificity of PCR results, as we've just discussed. The second property is processivity. Processivity is a measure of the number of nucleotides incorporated by the enzyme in a single binding event until the enzyme dissociates from the template. It is related to enzyme's affinity or how tightly it binds to the DNA template. High processivity of enzyme contributes to faster DNA synthesis, high inhibitor tolerance, and longer fragment amplification. While TAC and PFU were useful for many basic applications, these polymerases were still not very accurate. They were not very reliable, were also very slow, and had very small read length. The next big breakthrough in PCR came in 2003 with development of so-called fusion protein technology, by which DNA polymerases were fused to small DNA binding proteins. And the whole idea of fusing DNA polymerases with DNA binding protein came from looking at DNA polymerase processivity. Now, polymerases like TAC or PFU are able to incorporate only a very low number of nucleotides during the synthesis. In other words, they have low processivity. This is because they are loosely bound to the DNA template and they dissociate from the DNA strand rather quickly, and especially when they encounter the inhibitor. In turn, due to the low processivity, they are unable to amplify long fragments and they are slow. Now, it was known that naturally in our cells, so-called processivity factors bind to the polymerase, it increases processivity and allows it to stay on the DNA longer, for example, during the DNA replication. These processivity factors also allow polymerase to move faster and to overcome inhibitors present in the cells. Thinking about this, our R&D scientists fused thermostable double-strand DNA binding protein to the polymerase to mimic processivity factors naturally found in cells. And this fused polymerase indeed became highly processive, tolerant to inhibitors, and it was able to amplify longer fragments compared to TAC. And this is all because the double-strand DNA binding protein helps the polymerase to bind to the DNA tightly. It locks the enzyme on the strand and does not allow it to fall off quickly. In other words, scientists were able to create highly processive and inhibitor-tolerant polymerases. And one of them is fusion high-fidelity DNA polymerase. Here, scientists fused Pyrococcus species-like proofreading DNA polymerase to double-strand DNA binding protein from an organism that lives at very high temperatures. Indeed, this fusion polymerase is highly processive, 10 times more processive than PFU and 2 times more than TAC. It is very fast. It needs only 15 to 30 seconds for kilobase amplification because the double-strand DNA binding protein, it is able to generate long fragments of at least 20 kilobases. And as discussed before, high processivity helps DNA polymerase to tolerate PCR inhibitors. Fusion enzymes can tolerate inhibitors very well and therefore can be used in applications with samples containing PCR inhibitors or even with unpurified samples. Fusion polymerase indeed has a very low error rate. Less than 1 in 10 colonies may contain an error when amplifying 3 kilobase fragment with 30 cycles using the enzyme. Fusion is therefore widely used for many high-fidelity applications, including cloning and sequencing. Due to its other features, it is also used in many more difficult and non-typical applications, including long PCR, amplification of GC-rich templates, or even direct amplification from crude DNA samples or tissue samples. It also has a hot start version for applications requiring higher sensitivity. Since the fusion protein technology turned out to be very successful, scientists continued developing enhanced products and introducing new features for even better PCR.
Okay, that brings us to our second poll question here. Which of the following is a feature of DNA polymerase hot start technology? Option 1, method of amplifying DNA that uses a thermostable DNA polymerase. Option 2, it reduces non-specific amplification. Option 3, it involves the use of an antibody that blocks the activity of the DNA polymerase. Option 4, it is a technique used to increase the yield of PCR products. I will give you a few moments to select your answer. All right, and we have our last and final poll question for the day. Are you interested in getting a quote on fusion enzymes for your future research? I will give you another moment to select your answer. All right, thank you so much, everyone, for participating in these polling questions today. Marija, I will go ahead and turn it back over to you.
So as we discussed, PCR evolved significantly from its first predecessor in 1980s. The evolution of DNA polymerase, which of course was paralleled with evolution of thermocyclers, resulted in modern PCR, which is fast, robust, and more reliable, and allows us to streamline entire PCR-based workflows significantly. But there is still some space for further improvements. So what else can be done in order to improve performance of DNA polymerases? Certain polymerases do not require primer annealing calculation due to their novel buffer composition. This buffer is specially formulated to allow primers anneal at 60 degrees with high specificity. Here's how it works. Isostabilizing component present in the buffer ensures that primers, even with different annealing temperatures, anneal at the correct location with high specificity. This simplifies PCR workflow because far less optimization is needed and fragments can be co-cycled together. Universal primer annealing temperature brings a range of other benefits that simplifies PCR workflow. All primers anneal at single temperature, so you can skip temperature calculation. This simplifies reaction setup when working with multiple primer sets and enables co-cycling of different fragments at a single run, greatly shortening experiment time. Universal primer annealing also enables different length fragments co-cycling, so different PCR assays can be cycled together using the same protocol with universal primer annealing temperature and extension step of the longest fragment. So now, instead of running thermocycler 4 times, all experiments can be done in just one run. Here's an example where 4 fragments from 30 base pairs to 4 kilobases were co-cycled together at a single run with and without primer annealing feature. And just recently, original Fusion DNA polymerase was upgraded. Now, Fusion DNA polymerase is available with this unique buffer, which not only enables universal primer annealing, but also enhances overall polymerase performance. Fusion plus DNA polymerase does not require use of TM calculator, which was mandatory with the first Fusion version. It now has higher fidelity, more than 1 times TAC, higher sensitivity, better amplification of G series targets, and higher tolerance to inhibitors. With Fusion plus DNA polymerase, PCR accuracy is even higher, and this leads to even better experiment outcomes. In this example experiment, amplifying 3 kilobase size silicon, less than 1 clone in 25 may contain an error. So now the same Fusion enzyme became even better, has lower error rate, and also better performance in other high-fidelity applications. Now, one further development is modification of current polymerases. Uracil tolerance, for example. There are a number of applications which require amplification of uracil-containing DNA. Non-proofreading enzymes such as TAC can amplify uracil, as well as use DUTP in the reaction mix and incorporate it into a newly forming DNA strand. Proofreading polymerases are, however, inhibited by uracil in DNA template as well as by DUTP in the reaction mix. Proofreading polymerases contain so-called uracil binding pocket, which can bind uracil, and once uracil is bound, it blocks the extension. This is a very important mechanism of archaea by which they avoid amplification of damaged uracil-containing DNA. Because of that, in many cases, researchers use TAC whenever they have to amplify uracil-containing DNA. Here are some examples where there is a need to amplify uracil-containing DNA or work with DUTP-containing PCR mixes. In epigenetics, for example, for methylation analysis, final sample is analyzed by sequencing. For this application, one would need a polymerase that can amplify uracil-containing DNA accurately and efficiently. Then we have amplification of damaged or Asian DNA. Cytosine deamination occurs spontaneously over long periods of time and results in the accumulation of uracil in DNA. In many PCR applications, we need to amplify Asian DNA or damaged DNA for further analysis by sequencing, and for these applications, high accuracy is desirable as well. Finally, in order to prevent false positive results in PCR due to carryover contamination, DUTP can be used in PCR mix and then sample treated with UDG enzyme. In this case, we would also need tolerance to DUTP. So for some of these applications, such as sequencing or cloning, the use of TAC is not optimal because it lacks accuracy. Therefore, it is highly recommended to use proofreading polymerase that is tolerant to uracil. An example of such enzyme is Fusion U DNA polymerase. The enzyme was created by making a point mutation in uracil binding pocket. This mutation makes the enzyme tolerant to uracil. It can incorporate DUTP into newly forming strand. Fusion U, in addition to uracil tolerance, has all the same benefits as Fusion. So it is a high-fidelity enzyme, generates high yields, is a faster enzyme, and more robust overall. Here, in comparison to TAC, you can see much higher PCR product yield compared with Fusion U on templates containing uracil. One other use of Fusion DNA polymerases is direct PCR. Fusion enzymes are highly robust and inhibitor tolerant, which enables them to be used in direct PCR. Direct PCR allows DNA amplification directly from unpurified samples. A sample tissue is placed directly in the PCR reaction mix. Because no DNA purification is required, direct PCR can streamline PCR significantly. Usually, there are two protocols for this approach: direct protocol from sample to PCR in one step, and the dilution and storage protocol. It is more flexible because sample can be stored for some time and multiple PCRs can be done from the same sample piece. So with direct PCR, there is no need to modify sample. PCR is done straight from the tissue, significantly saving time and resources. It works with many different samples, whether it is plant, animal, or human tissues. It is mainly used in applications like genotyping, sequence detection, where you need to quickly screen for yes or no answer. So Fusion DNA polymerase has gone through many upgrades and developments that made it a polymerase of choice for a wide variety of applications. Since its introduction in 2003, there were at least 10 more new products developed, including hot start versions, Fusion U, Fusion Multiplex, and most advanced Fusion Plus DNA polymerase. And since the discovery of PCR, better and better solutions were discovered, such as Stack and PFU DNA polymerase. By intelligent design and fusion protein technology, we designed DNA polymerases that are faster, more specific, and more resistant to PCR inhibitors. We even started to design polymerases so that they work optimally for specific PCR applications, for example, Fusion U. Of course, the evolution of DNA polymerase was paralleled by the development of PCR instruments, and this resulted in a modern PCR as we know it today. The way we see PCR evolving forward, even more faster reactions, where it takes only several minutes, further reduction in reaction volumes, as well as miniaturization of PCR devices. All this will enable PCR to be used in clinics and hospitals, as well as in the field as an effective tool for medical diagnostics. Direct PCR analysis from crude sample material is something of interest as well, due to protocol simplicity and time to results. In Thermo Fisher Scientific, we are developing PCR tools, including enzymes, instruments, and consumables to further support PCR evolution and enable it to reach its next developmental stage. So thank you all for listening to today's webinar. And if you would like to learn more about our offerings, please visit thermofisher.com/fusion.
Thank you very much, Marija Navickaitė, for your very informative presentation today. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. All right, let's go ahead and get started. Question 1, is it possible to do multiplex PCR using Fusion?
Question? Now, I did not talk about multiplex PCR during this webinar, but Fusion polymerase can be used for multiplex PCR. Standalone enzymes can multiplex several fragments. And then there is also Fusion U multiplex PCR master mix, specifically designed for multiplex PCR up to 20 fragments.
All right, wonderful. Thank you. It looks like we have time for one final question here. Question number 2. Could you elaborate more on how universal annealing works?
That's a very good question. Thank you. Using standard PCR buffer, primer annealing must be very similar for them to anneal accurately. If the temperature differs, there is a risk a primer will not completely anneal to the target sequence or will anneal at the incorrect location. A PCR buffer that enables universal annealing at 60 degrees contains isostabilizing molecules that ensure primers always anneal at the right location and with high specificity. These molecules, they stabilize the primer DNA complex.
All right, wonderful. Well, thank you again, Marija Navickaitė, for your time today and for your very important research. Before we go, I would like to thank our audience for joining us today and for their very interesting questions. I would also like to thank our sponsor, Thermo Fisher, for sponsoring today's webinar. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.